So welcome everyone for joining us. I'm Terry Klinger at the University of Washington with Jan Newton. I'm the co-director of the Washington Ocean Acidification Center, and I want to welcome you to today's seminar. Our seminar is, this is the second in a seminar series hosted by the California Current Acidification Network, otherwise known as CCAN. We welcome broad participation from everyone in the ocean acidification community, and we're happy to have you with us today. So today we are going to be talking about the use of pteropods as a case study in water quality assessment for ocean acidification. Our seminar, as I said, is co-hosted by the Washington Ocean Acidification Center at the University of Washington. This center was established by the Washington State Legislature to promote and coordinate research on ocean acidification in Washington State. Our speakers today are two. First, Nina Bednarczyk, who is a research associate and science policy specialist here at the University of Washington. Her research focuses on the effects of ocean acidification on pteropods. She's worked in many different places in the world. One of the goals of her research is to translate and use the science that she obtains for the purposes of policy and management. Brad Warren is our second speaker. Brad is the director of the Global Ocean Health Initiative. This is a joint initiative of the National Fisheries Conservation Center and the Sustainable Fisheries Partnership. Working with coastal communities, seafood processors, and tribes, the Global Ocean Health Initiative focuses on expanding the toolkit for adaptation and mitigation so that the ocean can continue to feed billions of people. So welcome, Brad. Today's format is this. Nina's going to start with a presentation, sort of a data-driven presentation, in which she argues for the use of pteropods as a case study in water quality assessment to evaluate ocean acidification. Brad then is going to follow with comments on the risks to plankton and the focus that that puts on carbon emissions. I want to especially thank Laura Francis and Jennifer Mintz for their really capable assistance. We could not have done this without them. I also want to remind people that there will be a video archive and PDF of this webinar available. If you are viewing this now, you're probably already aware of that availability. And finally, I want to alert you to the next discussion hosted by CCAN. This will take place July 22nd of this year, and will concern ocean acidification monitoring. It will be presented by Jan Newton and industry partner Terry Sawyer. And as you can see there on your screen, registration and more information will be distributed via CCAN. So thanks for joining us again. And at this point, I want to turn the presentation over to Nina.
So I will try to demonstrate the use of pteropods for water quality assessment for ocean acidification in this talk. So let's start with the carbonate chemistry for the beginning. So really, if we want to understand biological responses, we need to have a solid grasp of baseline environmental conditions, and in this particular case, carbonate chemistry conditions. So I'm sure you have seen this figure before, but this is showing the timeline of ocean acidification on the US West Coast through the pre-industrial times, current, and by 2050. And what we are showing here is the percentage of corrosive, so undersaturated waters, and how it was developed in time, basically. The left panel, the pre-industrial, basically shows that during that time, pretty much all the water was favorable for pteropods, so supersaturated, with just a few blobs of near saturation states along the Oregon coast. But if we switch to 2011, we see that there are large spatial extents of undersaturation, especially happening along Washington and Oregon coast. And further, by 2050, we'll see that approximately 70% of these waters are becoming corrosive, and that definitely means habitat loss for a lot of species, including theropods. So by now we are all aware of the seriousness of ocean acidification impacts on ocean life, and there is a need to develop a water quality standards to evaluate the status of waters in respect to ocean acidification. And in particular, we need to develop this linkage between the exposure measurements and an effect to biological species or ecosystem. So a variety of different agencies have recommended the use of biological indicators, and this is really where theropods are fitting in really well. There are three criteria that are setting the requirement for the aquatic life assessment, and these are the following. So first, there has to be attribution of observed effects to ocean acidification. And we can either do this to correlative studies of field data and ocean chemistry. We can either do that in a lab and project the likely ocean acidification effects that will occur in the natural environment, or we can do that as a combination of a lab and field studies. The second criteria is that we have to demonstrate population level effects on the species. And we can either do that for the natural population or again in the experimental conditions where we have to demonstrate decreasing survival or fecundity as a likely population effect in the natural environment. The third criteria is demonstrated ecological importance. So if species get affected, that would potentially have an effect on the ecosystem level as well. So let's see these theropods now. These are a variety of different species that we can find in the California current system. Some are shelled, some are non-shelled. All are called theropods. In this particular talk, we're focusing on the upper left, Limacina helicina species. So they are pelagic zooplankton group, and they're basically present in all ocean basins along the globe. Here in particular along the US West Coast, they can compose up to 10% of total number of organisms in the upper 40 meters and this is the reason why they are considered an important component of North Pacific community, especially because they have really high grazing rates and can affect phytoplankton blooms. But even more importantly, and this is where that criteria on demonstrating ecological importance come into place, is that they represent a food source for a variety of different organisms. I have demonstrating just a few photos of a lot of species that are ranging from fish to birds and whales. And in the years of high theropod abundance, they can really represent quantitatively important food source for example, pink salmon, where it was discovered that approximately 50 to 60% of their diet can be composed of theropods. So when we start thinking about theropods as indicators and how to attribute observed effect to ocean acidification, we need to be aware of two things, two parameters. It's the magnitude and it's the duration of exposure that will really determine their vulnerability. This is especially important in the case of future OA conditions, which will be prolonged, more intense, and more frequent. So how will they respond to these conditions? So far, we have gathered several lines of evidence that suggest that theropods are already negatively affected by ocean acidification exposure in the natural environment. So what we have observed the changes were in shell dissolution, shell calcification, and in survival. And I will walk you through this. How we have come to realize all this was through the combination of observations, experiments, and modeling. And what I really want to point out here is that approximately 50% of all of our observations are actually done in the state waters. The blue line here represents the border for state waters. So we actually have quite a lot of observations from the biological experiments tied to the carbonate chemistry that can be used for demonstrating the effects of OA in the state waters. So it is really important that we understand the duration and the magnitude of exposure to ocean acidification in the species that we are observing. And in this particular case, modeling comes really handy. We have done this modeling exercise as tracking drifting particles. The duration for this modeling exercise was for 2 months, from early July to the end of August, during the upwelling season. And what we are projecting on this photo is we are trying to create some sort of visualization tool that will allow us to observe what is really happening with pteropods as they are basically traveling from the north to the south. And what it became apparent to us is that pteropods from the northern region, they are maybe exposed through this 2-month upwelling time 2 weeks to the undersaturated waters. And further down in the south, pteropods are exposed to undersaturated conditions for much longer. So really, pteropods from the central Oregon, for example, are much more exposed to ocean acidification stress than their northern counterparts. So I'd like to walk you through different responses that we have now observed through the experiments and observations in the natural environment. One of these is shell dissolution. And shell dissolution is really a very powerful parameter to observe because it very closely corresponds to carbonate chemistry conditions. And these changes on the shells are happening really quickly. It can start happening from days to weeks time, and we can see during one season, we can already see the effect on this. This is one of the severely affected pteropods found in the Hood Canal, and we see these big holes on the shell. And along the west coast, currently 1 out of 2 pteropods in the coastal environment show severe cases of dissolution, such as seen on this image. And we estimate that by 2070, approximately 70% of individuals will show severe cases of dissolution. And this is particularly found in the northern and central California current system region. So those areas are the ones that are particularly vulnerable to ocean acidification. We have also tried to quantify dissolution, and here we're showing the strong positive relationship between the proportion of pteropods with severe dissolution and aragonite saturation states. And so basically on the left, in supersaturated conditions, this would be pteropods from the offshore conditions with very small percentage of individuals with dissolution. And on the right side, we have pteropods from the onshore conditions, basically with very high percentage of dissolution in the near or undersaturated conditions. Basically, with the increase of the undersaturation extent, we're basically reducing the suitable habitat availability for pteropods. We are basically squeezing them out of their environment. So if dissolution is really the metrics for ocean acidification monitoring, because it seems to be a very straightforward tool for measuring that, this is what we have now learned about shell loss. So shell loss, as we see on this figure, is tightly correlated to carbonate chemistry conditions and exposure time. And if we follow the contour of, for example, 33% shell loss, so when 1/3 of their shell is dissolved, these kind of things can happen in the near saturated conditions when pteropods are exposed to them for approximately 2 months. But on the other hand, if they are living in the undersaturated conditions, these changes, as seen here, can happen on the 2-week basis. I'd like to point out that sometimes the undersaturated conditions in the US West Coast can really last for much longer, up to 3 months, and those are undersaturated conditions. And if we follow the yellow field, that basically means that pteropods on a seasonal basis can lose quite a lot of their shell, and in the future this might actually mean that even greater percentage of their shell is lost per season. The second tool that we are now developing that could potentially be used also as indicative of what's going on with pteropods in the natural environment is reduction in calcification. And again, as with the dissolution, shell calcification closely corresponds to carbonate chemistry conditions, and we can measure these responses again on a very short timescales. This is how pteropods look like from the natural environment. We have these three categories of responses. We might find pteropods that are calcifying fully, so to full extent, and this is here projected as a fluorescing fluorescence, to extent of the fluorescence. And so if they are calcifying, they will have full shell basically calcifying and fluorescing. Second level response is when they're fluorescing only at the growing edge. So they have reduced their calcification scope. The third line is basically reduced calcification or almost absent calcification. The best thing with this is that calcification closely corresponds to where we have actually found pteropods. So fully calcifying pteropods, fully fluorescent, are found in the offshore region. So where the conditions are supersaturated, so favorable conditions for pteropods. On the other hand, pteropods that are only calcifying or growing at the very last edge, so number one, are the categories found in a nearshore environment. But we see that in some of the really coastal environments, pteropods have lost this calcification ability and have stopped calcifying while living in the undersaturated conditions. The two videos that I will show now are clearly showing how dissolution is affecting pteropod swimming abilities, but can also tell us quite a lot about their impairment. So this is the healthy pteropod with intact shell and obviously not compromised by anything. It's a very fast responsive, fast moving pteropod. On the other hand, this is pteropod with very corroded shell, and this pteropod is basically passive, is not responding. It has lost to a large extent the swimming abilities, and it seems to be seriously compromised by dissolution. Once the pteropods are in a shape like this in the natural environment, they are also much more prone to infection and to predation, so another stress for them. This is the third level of responses that we have observed in the natural environment, and these are the changes in behavior, basically. Before, I haven't mentioned that pteropods are vertical migrators. During the day, they go down to 100 meters approximately, and during the night, they come to the surface. So we have studied this migration behavior in basically ideal settings in the eddy-associated front, and we had the onshore and the offshore station to compare the vertical migration in there. And two stations, onshore and offshore, basically differed in the depth of the aragonite saturation depth. In the offshore station, the depth was at 200 meters, and on the inshore, in the inshore stations, the depth was at 100 meters. So it was this 100-meter difference. And there we were basically observing through using the space for time approach how and if pteropod vertical distribution was changing, and if there are any differences in species richness as well. So this is what we have found out basically. In the onshore station, where there was a shallow depth of undersaturation, we see the missing layer of pteropod in that depth layer. And similarly, we can also see that missing the reduction in species richness in there. So we see these pteropod changes with the incurrence of the undersaturated conditions. But if pteropods are changing their vertical distribution, that most likely means that their immediate predators like fish might have to change their distribution as well. So this is already bringing in the ecological and potentially ecosystem implications into this. I'm not showing you the results of the individual-based model exercise that we have done, but I just want to point out that if pteropods started to migrate to much more shallow extent, they get advected to the near-shore environment much more quickly, and there they're even more exposed to these unfavorable conditions, and so ocean acidification. Another thing what we have measured was survival. So we were measuring the survival on the individual level, and we have been seeing the decline in survival. So here we are now trying to study multiple levels of biological organization. So we have been through the dissolution and classification of cellular responses, and now it's the individual response of the dissolution. So what we have been doing is we have been exposing pteropods to experimental conditions, to a variety of different high CO2, after they have experienced a variety of carbonate chemistry conditions for a few months in the natural environment. And what we basically see that pteropods living in the unfavorable conditions in the natural environment before had approximately 90% lower survival in the undersaturated conditions in comparison with the with pteropods that were put in the experimental conditions in the near saturation state. And another thing is that survival very closely corresponds to the aragonite saturation state. So we have this linear response. We see that the error estimates are quite big. However, that might indicate that there is some sort of acclimatization adaptation scope for pteropods that they might use for the survival. I think this exercise for the survival was very useful because it helped us identify survival thresholds. So aragonite saturation state in the natural environment that might put survival scope of pteropods under more light. We have found that near saturated conditions of approximately 1.1 as well as undersaturated conditions of approximately 0.9 are two functional survival thresholds for pteropods. So in the future, studies might use this threshold for examining the effects. So if all the processes are so well correlated to carbonate chemistry, the question was then, can we integrate across aragonite saturation states and see how these pteropods are responding? And here we are basically comparing how % of the solution found in individuals is impacting their survival. So what we are seeing here, that this dissolution might be very strongly actually affecting individual survival rates. So with with more individuals affected by dissolution, we might have lower survival extent. And this could be made as a very simple application for monitoring as well. Let's presume we take approximately 100 pteropods in the natural environment, and 2/3 of pteropods would show severe shell dissolution. We will basically move up the line and determine that we can expect approximately 50% survival only in the natural environment on the individual species. So this can be a very powerful monitoring tool that we can use, having combined and integrated all these biological responses together. So we have now demonstrated that we have the responses across the multiple levels of biological organization from the cellular individual levels. But there are some indications that these changes are happening on the population level already as well. So we have demonstrated short-term survival impact as well as how dissolution affects the survival. And with that, we have basically fulfilled the second requirement that is to show demonstrating decrease in survival or fecundity as likely population level effect. But we also have seen that some of the studies from the Northern California current system as well as Central Central California Current system are showing the long-term population decline. And this might coincide with the fact that the conditions, ocean acidification conditions in there are much more severe. On the other hand, pteropods from the Southern California Current system have not shown long-term decline. But we also know that the conditions in there are much less severe, and the region is mostly not undersaturated. Modeling work can, to a large extent, contribute to understanding of what is happening in the natural environment. We can use pteropods as a case study and as indicator testing. Atlantis, for example, as a model could be used for both scenario testing. And really that helps us understand how to parameterize, for example, pH-sensitive species within the model. What information would we need to gather from the empirical studies? How the different biological processes are linked together. On the other hand, if we are presuming that pteropods can be an indicator for the rest of the ecosystem, basically a warning, early warning signal of the changes that are happening, this is how we would use them as an indicator testing. So, as a summary of our conclusions or our results and the results that we have up to now, are showing that pteropods are really this ideal sentinel species, not because they are present everywhere and we started to understand the mechanisms, but mostly because we have now developed the methodological approaches that can be quite easy, straightforward, rapid, and cost-effective, and can potentially be used for monitoring of the responses to ocean acidification. I think the most beautiful part in all this is that their response to the carbonate chemistry conditions. It's very quick, it's very sensitive and specific. With the majority of other organisms, there might be a confounding factor of temperature, but it seems that the primary response in pteropods is to aragonite saturation state. As said, pteropods can be used as early warning signal, and that's really important because we can start detecting the changes before they actually occur on the population level, and for some of the species that might already be too late. There are currently some gaps and future research is needed predominantly to reduce the uncertainty, predominantly with also how to extrapolate our results across time and scales. And I think what we have learned is really that scaling up from cellular to individual level, that seems to work quite well. We have enough evidence that that is a robust application, but we need to establish stronger link between the lower and higher biological organization levels. And at least some of the tools available for us is if we want to study these lower level effects that are directly translating the effects on the higher biological organization levels. And in that way we can significantly reduce the uncertainty while doing this. I'd just like to acknowledge all the people that have been involved directly or indirectly in this project and thank them for their support and help. Thank you so much.
So thank you, Nina, for that. That was really helpful. I'd like to now invite Brad to comment from his perspective. It'll be just a moment as we transfer screens to Brad.
I'm happy to comment on that, and there's a couple of things that I'll discuss. One, I think that Nina's work is consequential. There is a track that applies to this problem, and finding ways to measure and what needs to be measured to apply the regulatory structure to it has been a discussion going for several years. this points to a path that may make that body of law more strongly applicable, and that will have effects. I suspect it will drive research money into the field. I suspect it will make a lot of people, maybe me, uncomfortable. It could polarize things quite a lot, but it's probably coming. So I'm going to switch to a full slideshow presentation mode here, and I'll talk about, I think the obvious implication here is that when you start seeing signs of serious stress on the plankton system that fish eat, if you're in fisheries, your options for adaptation now, anyway, look pretty thin, and that means your primary option is in emissions reduction, where you want policies that reduce the risk to these resources. That's been something people have regarded as almost hopeless for a long time, and I'm going to present some slides very quickly that make a case that, in fact, a lot of progress is happening, more than most of us would have thought. So I'll go to the next slide here and see. There we go. At the moment, after the last decade, we've seen very brisk growth in the development of cap-and-trade carbon policies around the world. Europe was the first in 2005, and it was a lonely experiment at the time. There are now 35 nations, 12 states and provinces, 7 cities, and a little bit more than is shown here that are involved in doing this. Collectively, they add up to about 40% of global GDP, and there are more coming. This is a map of places that have in force these mechanisms, using essentially a hard cap on emissions and then dividing it up into quotas, much like we use in fisheries, and allowing people to trade it, and that turns out to be an efficient and flexible, powerful tool. It's globally linked up and will become more so, and that's an advantage because you need to move really considerable amounts of money around the world to address this problem. Markets turn out to be one of the ways people do that, maybe the best way. So I'm going to go, before we leave this slide, I'll mention that there's one major country missing here, and that's this one here in the blue circle. That's India. They launched their own cap-and-trade system in 2011. At this point, that's a country with 1.2, 1.3 billion people, an economy growing at something like 5 to 7%. Getting countries like that, and China, where all these dots are over on the right, involved, and those represent provinces and cities in China that are currently operating cap-and-trade programs, which is a very clever way to do it. They started into this era using regional and city-based experiments to see how do we do this policy design, how do we make it work, how do we do it so we don't break the economy. They've clearly succeeded, and they're according to their plans next year to implement a full nationwide cap-and-trade in China. So that means India and China, the two great developing nation powerhouses, are already moving very strongly in this direction. Currently 10 states in the U.S. are involved in this, 9 on the East Coast plus California. Washington is considering going to a system like this. We'll see what happens. There are a bunch more of these systems that are scheduled to take place soon. Here we show the one over in China. There are others. And then there's some quite a lot more under discussion. I don't put a lot of credence in what's under discussion yet. That some things just happen, some don't. But the growth has been rapid, and I think it challenges the kind of tone of hopelessness with which we have approached this problem, and suggests that there's a track record that can be studied and learned from to evaluate which policies have been effective, how effective have they been, what have they done well, what have they done badly. We have quite an opportunity to learn from the design and experience of these programs. And it leads to some fairly obvious lessons that can be used in the U.S. and in other countries that are grappling with this. And it happens at a time when, for really mostly non-regulatory reasons, energy systems are shifting anyway. This can speed that shift to a lower carbon energy future. And it's an oddly interesting, dynamic moment. For all that we hear that it's hopeless and we might as well hide under the rug, people have been doing quite a lot, and a lot of it we can learn from and probably use. So I'm going to stop there and end with just a congratulatory note. I think Nina's work's very important, has consequence, and for fisheries, it means, for now anyway, that this kind of policy development could be very important. There are not many tools to reduce risk. This looks like the main one on the table right now.
Brad, thank you very much for that, for those comments and that analysis. I would like to thank Nina for presenting us with some compelling arguments to consider in terms of using pteropods as an approach to water quality assessment. Thanks, Brad. Thanks to all of you for listening in, and we look forward to your comments. I think that the presenters would be happy to converse with you by email if you have further questions to ask of them. I want to remind you again of the next CCAN conversation and webinar that's scheduled for July 22nd. That'll be Jan Newton and Terry Sawyer. And with that, I think we'll conclude this webinar. Thank you so much for joining us.